I thank the organizer for inviting me. Thanks, Natalia. And thanks for putting this great session together. And I have no financial interest to disclose with respect to this presentation. So when I prepared this lecture, I tried to see what kind of material is already out there, and it turns out there's a lot of great stuff out there. So I made a little YouTube playlist with my favorite lectures on layer fMRI analysis and acquisitions, including the ones from Saturday, so check it out. So increasing the spatial resolution with advanced instrumentation provides new insights. And I would like to show this with this little metaphor of astronomy, where we look at this beautiful night sky, and then you see that this part of the sky is just a bit brighter. And when you zoom in, you see it's a beautiful cloud. And when you zoom in further, you see there are subclouds. And you zoom in further and further until you hit the resolution limit of Earth-bound telescopes, like La Silla in Chile. And then with decades of hardware improvement going to space, you see way more, right? You see these additional contours and these little wiggles of the contours. And then with the newest generation of telescopes, you even see these little wiggles, like the dynamics of the clouds everywhere, right? And I like this metaphor because this is just about the time scale of fMRI development, where the big leap happened in the early 90s to non-invasively see activity changes in one area, but not another one. And then with decades of improvements and 2 orders of magnitude in resolution, you could also not only see different areas or systems, but also kind of how the activity is distributed within those areas. And with the newest generation, you can even see, like within the gray matter ribbon, how you resolve these subpopulations of neurons, these individual stripes that we believe refer to different cortical layers. And this is exciting because these layers, they represent different stages of neural processing. So you can activate them differently and learn additional information about neural processing. And I would like to make this clear with this little experimental series over there, where we just ask participants to do a standard tapping task, and at conventional resolutions, you have this kind of big blob. And then when you ask the person to just think about moving their hand, the same areas kind of seem to be activated, right? It's weaker and the blob is a bit skewed differently, but overall it's kind of frustrating how it looks so similar even though the behavior is completely different. Now, when you zoom in to this tiny patch of the cortex and increase the resolution further and further and further, you ultimately see that there are these different sub-distributions within the cortical ribbon. For example, these voxels here at the cortical surface, which we believe are mostly representing cortical integration for motor planning areas or sensory feedback, as opposed to the output, the deeper layer groups that trigger the muscles to move. Now, at these kind of resolutions, you can also look at the imaginary tapping, and for this kind of threshold, you see that it's kind of orthogonalized now in the sense that the deeper stripe is missing because there's no output happening, which I find really exciting because now we can kind of see how the same brain area is involved, but we see how it's doing different things. And of course, these kind of input-output circuits are only one specific application of layer-dependent fMRI, and it can be used for many, many more neuroscience questions. For example, at this conference, looking at attention modulation, or looking at predictive coding and prediction error estimations, or looking at how consistent the depth-dependent activity is in a given column compared to the neighboring column, or looking at directional connectivity, functional connectivity, or using layer fMRI as a kind of vascular filter to learn more about your conscious mechanisms and how it's different from macrovessels or parenchyma, as well as many, many more applications. To do layer-dependent fMRI for those kind of research questions, however, we do rely on decades of methodological improvements. For example, when you do want to do a layer-dependent fMRI experiment, you need advanced hardware, and we learned about this from Professor Feinberg already on Saturday. And then you need advanced sequences, which we learned from Professor Poser. Then you need to run your experiment, do your analysis, and draw your scientific conclusion at the end. And each step here is kind of built on decades of development, segregated quite now a bit, and really over-engineered. So you cannot simply kind of just change the field strength or the hardware piece at the beginning, or change the resolution and assume that everything further downstream is the same. So for example, when we take the most simple case, like an EPI, you go through k-space back and forth and back and forth. It's the most simple geometric pattern you can imagine, right? My 3-year-old understands this, but still, when you go to the scanner and you want to do this simple pattern, you are confronted with dozens and dozens and dozens of parameters that are interdependent and need to be optimized for layer-dependent fMRI, which is a challenge, even for the most simple things like EPI. Because like while EPI is the most efficient method, it's still not efficient enough to really capture the entire matrix sizes that we need for layer-dependent fMRI. So usually people need to subsample and skip case-based lines and then fill in these missing case-based lines by other means. And one popular approach to do this is, for example, GRAPPA. So in this toy model, when we have a time series of subsampling by a factor of 2, without filling in those missing case-based points, we would have aliased images. And then with GRAPPA, at the cost of a square root of r SNR penalty and a G factor, we can unalias them. Alternatively, though, you can use the same trajectories here with a tiny little different prefacer and combine two of them to not unalias them by GRAPPA and fill in the missing case-based points by so-called segmentation, which has less samples, accounting for the kind of square root of r SNR subsampling penalty, though the G factor penalty is something you really never get rid of in GRAPPA. So in the purely thermal noise-dominated regime, ideally, theoretically speaking, segmentation would always be more efficient than GRAPPA. In practice, however, it's not so easy because there are these shot-to-shot phase variants and variations. So for a conventional protocol of, like, let's say 0.8 millimeters, you can use the same kind of echo spacing and the same trajectories and reconstruct them in different ways. For example, doing a 6-fold undersampling, not doing GRAPPA at all, but just segmentation, which ends up being very inefficient and some physiological noise dominated. On the other hand, you can also do a lot of GRAPPA, but no segmentation, which puts you really in the thermal noise dominated regime. Even though you have a lot of TRs, they're just noise. So often you are kind of you have your optimum somewhere in the middle here, where you do a bit of GRAPPA and a bit of segmentation if you need to. Now, when we push this to the more thermal noise dominated regime of higher resolutions, you see that the optimum kind of moves towards the left, where we get most TSNR efficiency, like TSNR per unit of time, when we do very little or no GRAPPA. Now, when independent of all these sampling approaches within plane, there are also these different flavors of sampling across plane for layer-dependent fMRI. And we learned this morning, for example, in the early sunrise lectures, that at conventional resolutions, most people are really happy with 2D multiband or SMS sequences. At layer-dependent resolution, that's a bit different, and more people seem to prefer 3D readouts, specifically looking at like 95 papers that have been published in human layer-dependent fMRI since 2020, where there are almost double the number of 3D papers compared to 2D papers. And anecdotally speaking, I think 3D EPI was mostly developed for layers and high fields, because in the early days, people just could not get 2D to work with all these off-resonance effects and the shitty gradients of those days. Nowadays, I think 3D EPI is mostly popularized or became the standard because of the higher sensitivity, maybe because of the more flexible sampling patterns and sharper slice selection, but I think it's overall the sensitivity. Note, though, that for 3D EPI, it's a multi-shot method, so you are more sensitive to kind of non-linear field changes. For example, when you do very challenging tasks, like, for example, having people drink inside the scanner and do swallowing, you see that for 2D, while being maybe more salt-pepper noisy, the kind of center of the brain looks all right. It kind of moves up and down non-linearly compared to the rest, whereas 3D EPI is basically just unusable over there. For most of the time, people do not have those kind of challenging tasks and are kind of happy with those kind of protocols, even though they are not perfect. So, for example, when you look at it very carefully, you do see that there are these kind of low spatial frequency shadows over there, right? Which are not to be confused with off-resonance effects, but we believe they are rather coming from gradient imperfections and eddy currents. And for example, you can completely invert them by just switching the redirection and then using that to calibrate, or using those two images, complex valued averaging, and then get nice artifact-free images. And if you want to hear more about those fuzzy ripple artifacts, as we call them, come to the Thursday morning lecture. Now, independent of all these data sampling approaches, layer fMRI acquisition is still also limited as conventional fMRI by the fact that we are measuring neural activity indirectly. So we do rely on the neurovascular coupling, indicated here as an astrocyte. We have firing neurons, and there's an artery going through here. So when there's neural activity increase, the arteries are triggered to open up, increase their volume, which allows more flow to go through, and a short while later also the venous vessel here has an oxygenation change, which kind of shows the problem already, because this vein comes from somewhere, right, outside that voxel. It might drain neural activity as far away as 3 millimeters, which is particularly challenging for contrast mechanisms that are dominated from extravascular defacing, for example, like gradient echo bolt. And there are decade-old debates on potential auxiliary methods out there, and this debate is not solved and maybe never will be. But when you look at kind of 200 human layer-dependent fMRI papers out there, about a quarter of them is using gradient echo bold and just hopes for the best. About a quarter of them uses gradient echo bold and discusses other methods that might be more or less advantageous in different experimental setups, and about half of them is using other or a combination of gradient echo bold with other methods. And these other methods could be spin echo or grace or vaso or vapor, which all have their different kind of fingerprint for given experimental setups, like being really sensitive to the microvascular components or being less sensitive to microvascular components, but more sensitive to, like more generally sensitive to extravascular components from large vessels. And I do believe you can think about the spectrum of all these different methods out there as you would think about a spectrum of different vehicles. There's clearly no winner, right? Which one is the best? It depends on your experimental setup, and you want to tune and optimize your experimental task design and everything that you really benefit the specific method that you want to use at the end. And very often I do feel challenged by sensitivity and feel inclined to use gradient echo bold, which kind of makes me feel like this guy that takes Lamborghini to McDonald's because they worry about food prices. Like we invest so much effort and mental energy in efficient sampling, so we should be allowed to use that good sensitivity for good use. And hopefully Professor Kim will chime in on this discussion at some point. Aside of all these acquisition challenges, Natalia also asked me to talk about analysis, which I think is an important aspect in layer-dependent fMRI. Because layer fMRI, different to conventional fMRI, is kind of messy. It's not standardized just yet. And we have like messy artifact-written data. And when you look at this conference, as well as the last 3 conferences, I think the most common approach to do layerification is using a software suite called LANY, which is currently maintained by Farouk Gulban and supported by Brain Innovation, which basically gives you layers by any kind of segmentation you give it. It can be 2D, 3D. It doesn't need to fulfill topology requirements of closed surfaces without holes. And it gives you then, based on the segmentation quality, equidistance or equivolume layers out of it. Independent of the layerification, also columnarization is getting more and more popular. Basically zooming into a patch of the cortex, spanning a local coordinate system within that patch, and then flattening it to look at topographical representations. Like, for example, in the motor cortex here, looking at mirrored finger representations. Also, very exciting and online since yesterday, I think, looking at columnar maps of axes of motion in the human MT V5 from Alessandra Pizzuti, basically taking this tiny patch, flattening it virtually to a petri dish, and then looking at the interspaced axes of motion columns and how they spread across cortical depth. Another approach that is being more and more looked at for exploratory purposes are deveining models, where you try to mathematically parameterize your venous leakage, for example, by means of an asymmetric point spread function, and try to get rid of that by, for example, deconvolution or inverting the model and tease out these tiny microvascular signals that sit on top of this huge macrovascular signal in gradient echo bold. Other approaches are more tuned towards the kind of acquisition physics, for example, considering that when voxels have large straining veins, they have a frequency or phase difference due to activity, and that time course should be slightly different compared to the microvascular response, so you can use temporal phase regression to get rid of large straining veins effects and even blooming effects from PL vessels into the upper layers. When it comes to motion, when it comes to high resolution, many people worry about motion. I don't too much. It's annoying. At the end, it's still fMRI, so motion is never really constant. So yes, we lose TRs and we lose some participants, but at the end of the day, for the neuroscientific studies that we are aiming for can live with those kind of dropout rates. And if you tweak the post-processing tools that exist already for layer-dependent fMRI, I think you can always get kind of acceptable quality of motion across runs. A far bigger challenge for layer-dependent fMRI analysis, I think, are tissue type segmentations. In my experience, naive experience maybe, there are no clear software tools out there that provide high-quality tissue type segmentation in the EPI space across brain areas. in some brain areas, gray matter is just too thin, and sometimes sulci are completely missing, and it easily takes up to 60 hours of manual corrections of people. And here, poor Kenju even has a wristband, which is, you're laughing, but it's quite common when you walk down the hallways of layer-fMRI labs that just students sit there and correct and correct. There is a YouTube tutorial from Farouk Gulban showing tips and tricks and really trying to optimize and make these manual or semi-manual adjustments of automatic tissue type segmentations more efficient. Check it out. One approach that is very popular in layer-dependent fMRI analyses, and I think almost half of the layer-fMRI abstracts this year use it, is the patch-wise removal of principal components that look like Gaussian noise, aka Nordic from Luca and Steen, which can tease out signals in even very, very noisy data. Here, for example, even at 3 Tesla layer-fMRI VASO for this visual motor paradigm where you barely see something in the visual cortex, definitely nothing in so much as sensory areas. And then with the inherent estimation of the noise floor and thresholding in Nordic, you can tease out much more convincing activation patterns as well as the sensory areas. If you get really greedy and remove more and more and more components successively to even get these tiniest signals in the primary motor cortex, ultimately you do lose your interpretability of the effect size and spatial pattern, though. In cases where you can assume that layers or that neighboring columns are doing the same thing, you can also do layer-specific smoothing to deal with the noise level of individual runs, as it has been implemented in many software suites now. Aside of these aspects of acquisition and analysis, I think there are further aspects in the field of layer-dependent fMRI that we need to pay attention to make the field of layer-dependent fMRI successful. Because at the end of the day, you can get the right neuroscientific answer independent which kind of method you're using for acquisition or analysis. In fact, one of the most reproduced feature in the field of layer-dependent fMRI, the double hump in the primary motor cortex, you can see it independent of the method with CBF, CBV, vapor, gradient echo bolt, with phase regression, GRACE, or spin echo EPTI. No matter if you do kind of vertex-based or voxel-based layerification. Now, one of the least reproduced features in layer-dependent fMRI are simple things like attention effects in V1, where some people kind of see middle and superficial layers, some see upper and deeper layers, some see kind of a dominant effect in the superficial layers, some see an effect across all layers identically, some see deepest layers only, and some see most significant effects in the middle layers. And all of them use gradient echo bolt, and not almost all of them use gradient echo bolt. All of them use bolt, though, with vertex-based layerification. So I think it's not really the method that is challenging only, but I think we also need to pay attention on how we take our very specific tasks with the specific modulation conditions, with the specific control conditions to generalize to these big concepts like attention. To give you a feel where the field is going and where we're coming from, let's look at some statistics. So it took quite a while for layer-dependent fMRI to kick off. It's an old field. In about 2015, it started to kick off, and I'm really happy about the development that nowadays there are much more application studies than these methods, purely methods-focused studies. I'm also happy about the development that it's expanding across brain areas. It's not vision anymore only, but there are more associative or cognitive high-level orders areas looked at too. I'm also kind of happy of the development of data sharing, where until 2019, almost nobody made their data publicly available. Since 2019, it became more and more the standard, and at least 1 out of 4 papers makes their data publicly available. But still, we have a way to go there, I think. Other aspects where we do have a way to go are diversity. So we are really dominated by Siemens, which might not change in the near future, but also looking at geographic diversity. There's no Africa, and also, like Asia and America having most of the ultra-high field scanners, but still most of the layer developments are being done in Europe. And maybe even more alarming is the gender diversity or lack thereof, where only 1 out of 4 layer-fMRI papers is coming from a female author. And when you look at all the references in layer-fMRI papers, only 1 out of 7 references goes to a female authored citation, which is something that we as a field need to work on, because we are really performing worse than other subdisciplines of neuroimaging. Independently, of course, we also need to worry about our climate footprint. Like, just for this little session to get the academic speakers here emits more CO2 than it takes for a household for 2 years. And we learned yesterday from Professor Merkle that having a 7 Tesla just sit there consumes more energy than 60 to 100 households which is a lot. If you want to engage in the field of layer-dependent fMRI, please do so via, for example, the layer dinner group, the Laminouts, for example, by downloading an example data set and show the field how you get from raw data to your layer profiles at the end. This Twitter handle tries to keep you informed about ongoing events and papers. For example, there are 2 very exciting workshops coming up later this year. There's a 4-day educational workshops on layer-dependent fMRI in Boston at MGH, and there's also a 2-day workshop later that same month as part of the ultra-high field workshop in Minnesota. It's a hands-on workshop. And with this, I thank all my colleagues and you for your attention.